I'm Gary Peng, a research scientist with the Agriculture and Agri-Food Canada in Saskatoon. And my work is mostly on canola diseases, including blackleg and the clubroot. Often the most significant damage is we call the basal stem canker. That's when the infection started early, mostly from the cotyledon stage, and it grow into the base of the stem. And often they either girdle the stem and to destroy the whole plant completely, or it affected the nutrients and the water uptake, stress the plants. If you have a higher temperature drought condition, that will have even more yield impact to the crop. So that's how the disease affected the plants. Since 1990, we've introduced a couple of several varieties. Started with the two major resistant genes, and those are RLM3, RLM1, and gradually those genes are being incorporated into more and more varieties, and pretty much we have a large portion of the cultivars carry those genes. But the pathogen adopted to this resistant gene, and in the current pathogen population, and those genes are no longer functioning as the resistance genes. But at the same time, we still have a quantitative resistance in the background that seems to be doing the major lifting of the resistant work in most of our canola crops. The first part was the initial, some of results available prior to that it wasn't as conclusive because of a low-level disease happened in those trials. Secondly, was fairly limited because of the geographic location was mostly in central Saskatchewan. So what we wanted with this study was to have a multi-year, multi-site trial and test all the product, and hopefully we'll have at a higher level of disease to be assessed on, and also have different environmental conditions during the crop season, and to be able to assess the performance of those treatments better. And also we took all registered products for this disease and tested all of them in those trials, and to be able to get a bit more conclusive the information on this issue, and that's the reason for this study. The resistance, the moderately resistant cultivars are really representation of our current cultivar. Most of the cultivars are being grown there. And on those cultivars, the fungicides are really not necessary, and mainly because of the level of resistance they have. Now our data clearly indicated the level of a disease, blackleg, happening on those cultivars are rarely reaching the level the fungicide will provide any yield benefit. So I would not recommend any fungicide treatments on those cultivars. Although we don't quite understand why some of the fields are more severely damaged by others, even with the same cultivars. It's isolated cases. It may be related to some the events specific to that site. For example, the early hail damage cause wounding on the plants, and some are the insect related the damage, like early insect feeding on the cotyledons, which facilitated the infection there. Those things are still being investigated, and And what we can say for now is probably the fungicide can be used after early hail damage if crop can be salvaged, and that may be the place you can put a fungicide on. Or you are seeing clear trend of increasing level of disease despite the fact that you're changing the cultivars, you are extending the rotation, and you're still seeing the increase of the disease. So that means your risk level is increasing. That's probably when the fungicide can be considered for the early application. We don't have a lot of choices of fungicides in terms of blackleg control, but there are two chemistry. One is a strobilurin type, but one is a triazole type. Strobilurin type of fungicide seem to be slightly more consistent in terms of performance, especially relating to the yield benefit. Among the strobilurin type of fungicides, there are not a lot of difference, but triazole products seem to be slightly less. But but timing is also important. That when you apply on the heavy disease pressure and the application need to be made earlier. We use a 2 to 4 leaf stage for application. That seems to be a lot more effective than applying later at the early bolding stage for blackleg control. There are two parts. And one is that if it's not economically warranted, why do you do that? You don't get the yield benefit, especially when the disease level is not that high. And the second reason is that we have a very limited choices of chemistry, and especially the strobilurin type of fungicides are very easy for pathogen to develop a resistance on. So we need to preserve the product for the time we really needed it. So for that reason, I would recommend not to use the fungicide if it's not warranted. And sometimes the resistance level is a little bit a tricky thing to determine. But one of the things that growers can do is by harvest time, after swathing, just check on the incidence of a blackleg on their crop by checking 50 to 100 plants, those stubbles in their field. If they don't have the incidence over 10%, that means probably the variety is still doing okay. And most of the growers will rotate the variety to another company's product, and in their rotation, that is a good thing, especially with the potentially new varieties coming into the market and that carry different type of resistant genes. But even that, some of the scouting will help to determine what kind of a disease pressure they have on their farm, and that gives them the sense of what the risk level they are in, the disease is at.